Hello, everybody, and welcome to this episode of Genomics Lite. My name is Sam, and it's lovely to have you all with us.
And my name is Jack. And Sam and I both work in the Wellcome Connecting Science engagement team based on the Wellcome Genome Campus in Cambridgeshire.
And if this is your first Genomics Lite before, then welcome. If you're returning, then welcome back. But these sessions are basically short sessions that aim to explore the science that happens on our campus. And our campus is all about DNA. And this episode, we're going to focus on a couple of people who have been involved in one of our big research projects, the Human Genome Project. And we're going to talk a little bit about that today and try and answer the question, whose genome was sequenced first?
This video is the first of a four-part series of Genomics Lites, each one exploring a different big question. And you can find all the dates for those live streams and after those live streams, the episodes on our YouTube channel. We're going to be doing one every Thursday lunchtime through August. So our first topic, whose genome was sequenced first? Sam, how are we going to tackle this one?
Well, yeah. So as I mentioned, we've got two wonderful guests. We've got Nancy and Liz, who we'll be introducing in a little bit later, and they're going to take us back in time. They're going to take us back to the 90s when the first genomes were being produced on our site. But before we do that, let's get everybody up to speed about some of the things that we're going to be talking about. So let's break down our title a little bit, which is whose genome was sequenced first. So Jack, tell me a little bit about what we actually mean when we say a genome.
Yeah, great. So I know that you might already know all about this if you're watching, but let's make sure everyone's in the same room. So a genome is an organism's complete set of genetic instructions. That's the information that that organism needs to build itself, to grow, and to live. Now, most life around us, and I'm looking out of my window out here, I can see a lovely hedgerow out there. Most of the life that we can see around us is made up of lots and lots of cells, millions and millions of those cells. And each one of those cells will have a full copy of that genetic instruction, that genome within it. So you could say that the genome is the full DNA instructions in one of your cells.
Amazing. So another word that we've got in there is sequence. So that's a bit of a strange word. What does that mean?
Yeah, so sequence is one of those words that people who work in genomics or in DNA science use all the time, and we kind of forget that it's not a really common word. Sequence is the way that we describe a section of DNA code. So that DNA code that makes up the genome is itself made up of four different chemical molecules that we call DNA bases. Those bases are adenine, cytosine, guanine, and thymine. And to save some time, we just refer to them by their first letters. So A, C, G, and T. The order of those four bases in your DNA is what gives your body the instructions that it needs. And so a different order or a different sequence of bases will give a different instruction. ACT-GGC will give a different instruction to ACT-TCA, for instance. So those short sections are themselves DNA sequences, but your whole genome sequence is all of the letters in that genetic instruction book, all of those letters in the right order. And for complex creatures, that can be millions, usually billions of letters long, all in the right order. Pretty tricky to do.
Okay, so we've got genome sequence, and we've thrown a few dates around already. We're saying the 90s and stuff like that. Take us a little bit back in time. Give us a little bit of a history about when we actually talking about what's the timeline of all of this.
Yeah, okay. So this episode is about whose genome was sequenced first. So when was that going on? Well, genomes and DNA, that's been the way that life's worked since the very beginning. So those genomes have been there for billions of years, evolving, of course, as they go. But we as scientists only knew about DNA about 150 years ago and was only able to read that DNA from about 50 years back. So it's a really recent science, this actually. Now, what that means is that when we're talking about the early genomes that were read, that was happening in the 70s, these things called bacteriophages, which are viruses that infect bacteria, they have pretty simple genomes. They're only a few thousand bases long. As we progressed from the 70s, we got better and better and better. I'm saying we here, taking some credit, of course, a whole realm of fantastic scientists working on this all around the world. And so by the 90s, we were starting to do genomes that were millions of letters long. These were more complex things. They were bigger bacteria. They might be simple fungi, as we'll hear from Nancy, and into creatures that we might start to recognize, some very small worms, into flies, and then finally in 2003 the first human genome was published. So that's going to be the real crux of our episode here. We're thinking the kind of the 90s and the early noughties.
Okay, amazing. Now, final bit. Okay, who are we talking about? So the Human Genome Project, there's the human. Who is that person? Who is the person that is involved?
Yeah, so you're asking me for the answer for the whole episode here.
Exactly, yeah.
but so let's get it out there. So first off, if we think back, the 70s, 80s, 90s, those experiments that they were doing with things like worms and flies, they can grow those in the laboratory. So we've not got a problem with where we're getting these samples from. Once we get to humans, obviously, of course, we can't do that. We need to think about who we're getting our samples from to sequence the DNA. For the first Human Genome Project, published in 2003, those samples came from volunteers in America. And when they collected those samples, they anonymized the DNA that they took. So in theory, if we wanted to finish our episode here, we could say, we don't actually know whose DNA was in that first Human Genome Project.
Well, that would be an incredibly short episode, so I'm hoping you can delve in a little bit deeper for me and tell me what can we start to unpick a little bit from the information we are allowed to know about this.
Yeah, of course. So there's loads more interesting stuff that we're going to say. Don't worry, we're not finishing early. The first thing to say is we can say a bit more about who it was who gave those samples. So these were collected from an initial cohort of 20 volunteers in a town called Buffalo, which is in New York State. But if, like me, you're not very good with your American geography, Buffalo is in the northeast part of the US, just on the border with Canada. So anyone who's been to Niagara Falls will have probably been to Buffalo City. That's just where it is. These volunteers, they will have come along to this center in Buffalo that have put out an advert, and they'll have donated a little bit of their blood with someone called a genetic counselor. We have these in the NHS as well. They're people who are trained not only to know about DNA, but to be able to have conversations with people about their DNA and perhaps any ethical issues around it. So they will have given their blood to that genetic counselor, and they'll have talked through the consent of it being part of this research project. Now, how they were consented is a really interesting topic all of itself. I don't think we're going to have the space to really dive into it on this episode, but there's an amazing article all about it on this site called Undark. We'll post it underneath the episode when it goes onto YouTube. But once they'd been collected, those 20 people, they were then passed to some scientists with the names removed. Those scientists did a lottery-like process, is how it's described in the research paper, to pick out what they said at the time was 2 of those samples from 20. So the idea was that they didn't know whose samples from the 20 had been selected, even if they might have known some of the volunteers. Now, over the course of the rest of the Human Genome Project, some more DNA got added in, and some of that was not quite as ethically sound, I don't think. They said that one of these was from one of the researchers working on the project. There was another that they suddenly realized, oh, we need a bit more of this DNA, so it was collected from a family member of someone on the research project. Again, it's all in this Undark excellent Undark article, so you can have a look in there. But what we ended up with purposefully was what would be called a bit of a mosaic. Not just one person's genome, but actually a few different people's genomes pasted together. And we'll talk when we get our guests involved about why you might want to do that, actually, rather than just having one person from start to finish. But the other thing to say is that while the scientists aimed for that mosaic to be really different people, maybe each contributor only given maybe 10% of the human genome, actually that's not how it worked out. So about 75% of that original human genome that was published was just one person's DNA. We know that they were an African American person from Buffalo. And that's as much as I can tell you. The rest were some more of those volunteers and some of those other contributors that I mentioned.
Okay, amazing. Well, I think we have kind of started to give a really nice overview, and the most important thing is to start to get to hear from our guests. So I would like to introduce our first guest, who is Nancy Holroyd, who is a samples lead and manages all of the samples on this amazing project that I'm sure we'll get to hear a little bit about in just a second, the Tree of Life project. Nancy, you originally came to our site when it was first founded in 1993. So first of all, tell us a little bit about what you do right now.
Thank you. Yes, it's really nice to be here. So now I lead a small sample management team in the Tree of Life program, and we're responsible for bringing in all of the samples from different animals, plants, fungi, into our program to be used for the genetic research to sequence their genomes. So we ensure that those samples come in the right format and with all of the right information to enable the science to happen.
Amazing. And as I alluded to, you've been sort of around our site and then been on our site for a really long period of time, which is amazing to have your experience with us today. So take us back to when you were first handling those samples. What was that like? What were you doing back in when you first entered the Sanger Institute, or the Sanger Center when it was known then?
Yes, so when I first joined the Sanger Centre, it was originally back in '93. I was working in a small team, again, this time working on yeast genomes. And we actually, because it was very early on relatively early on in sequencing days. So the role that I had actually was included a lot of different tasks. Over the years, I think that as the genomics projects have scaled up, it's become much better for individuals perhaps to specialize in certain parts of the process as you want to scale up like a production line. But actually in the early days, it was quite an all hands on deck type of approach. And so when I first started, I would be culturing samples and extracting DNA and also doing some computer work. So it was incredibly varied, and it enabled me to have a chance to actually try my hand at quite a lot of things and be involved in very many aspects of the work.
Amazing. And just tell us how you link to the Human Genome Project, because obviously you were part of a team that was looking at something slightly different, but tell us how that sort of linked together a little bit for us.
Yeah, sure. So I wasn't actually part of one of the Human Genome Project teams. I was in the pathogen sequencing unit, and we primarily worked on bacteria or protozoan genomes. So looking at the DNA sequence of lots of organisms that cause diseases. However, the underlying technology for sequencing those was pretty much the same as the technology needed and being used for the Human Genome Project. So as an institute, there were a number of parallel projects going on, and we were able to really utilize, I think, the expertise in a number of different groups to brainstorm and think, how can we use this technology? How can we use different approaches to better help the Human Genome Project? Myself, I actually used to come in at the weekends to load sequencing gels. So although I wasn't part of the Human Genome project per se. I contributed to getting those samples on the sequencing machines. So it felt really good to be part of that collective effort.
Yeah, amazing. And you, we've spoken before, but you mentioned to us that you joined the site at 93. You hadn't gone to university or anything before that point, and then you went away. Is that right? And then when did you come back?
Yes, so I did join in 93 when the Genome Campus was really just starting out. And later on that year, I decided that I really did enjoy science, but I had an opportunity to go and study for a zoology degree at Imperial College, and that's what I went off to do for 3 years. But I was really fortunate that I was able to come back during Easter and summer holidays to earn a little bit of money, which is always useful when you're a student, and really keep my hand in with the genomics world. So I felt really privileged to do that. And when I left university, I kind of stumbled back to Sanger. I had no intention of coming back here. And when I did come back here, I had no intention of staying for the length of time that I have. And I'm still here. That was decades ago now.
Well, it's wonderful to have you, as we said. I'm going to hand to Jack to introduce our next guest.
Yeah, so I'm going to be bringing Liz into the conversation now, so Liz is a next part of our story, but fits really wonderfully together with Nancy. Liz works as a team lead in our sequencing operations, and I'm going to ask you to tell us a bit more about that in a second, Liz. But just to link this nicely together is, of course, we've got our samples coming in. And we're going to get onto those human samples used in the Human Genome Project, but that we can't just get them on our site and get to the DNA sequence in one go, can we? We need lots of different things happening in between here and there. So, Liz, tell us a bit about what you do and how that bridges the gap from sample to DNA sequence.
Certainly. Thank you for inviting me today. It's good to come and talk with everyone. So what my team do, I'm a team lead for a team of people who create libraries. That's a technical term, but what it is basically is we make that DNA within the sample able to be read by the sequencing technologies. So there are different reactions that we have to do, and the different stages of that process, my team will be involved in doing those. And then at the end, we'll have a sample of libraries which are ready to go onto a sequencing instrument so that then their genomic sequence can be read.
If we were doing the Human Genome Project now, what would it look like, Liz? We get these samples from Buffalo come in. What would arrive in your laboratory, and then what would be going out to the sequencers?
Okay, so there's one step before my lab as well, and that's a step where the samples are received, similar to the sample management that Nancy will have. And then also do some preparation work. So we can receive those samples as already extracted DNA. That means that some tissue or some blood, someone has had that from a participant and taken the DNA out. So then we can use that to make our library. Another way that it can come to us is as tissue or blood, and the sample prep team here can take that DNA out. So there's two different methods. Either they come already extracted and ready to go, or they come as tissue and blood and we can extract them and then hand them to my team to be read.
So you might get some kind of little tube or something like that. In fact, I've got a tube here. You might get one of these.
One you prepared earlier.
Tube. Yeah, one I prepared earlier. Yeah, I don't normally have these in my office at home, but yeah. So you might have a little Falcon tube like this with a sample in. Some team before yours, Liz, will do something to that to get the DNA out of it. Into your laboratory, you get a much smaller tube with a much smaller amount of substance in. Yeah, and then that you do some things with that to send it off into the sequencing.
That's exactly right.
Now, what would that have looked like back in the 90s, as they were beginning to build up the work for the Human Genome Project. It was a bit different then, wasn't it?
It was very much different. And in fact, when we spoke earlier, Nancy, you have a bit more experience with those backs and yaks, as they used to be called. I don't know whether you wanted to step in and help with that side of things.
Absolutely. So a lot of the sequencing that's done today is essentially breaking up that genome that is extracted from the samples, from the cells of the sample, into many reads, and the whole genome is sequenced effectively in one go. Lots of little bits of sequence from that one genome, and that's called whole genome sequencing. But in the early days of the Human Genome Project, something called a BAC, a bacterial artificial chromosome, or a YAC, a yeast artificial chromosome, would be made. And they sound like very complicated terms, but essentially, because for the human genome, and you mentioned earlier on that whilst it's very easy to get fly samples and yeast samples in the lab, it's not very easy to get lots of human material. So the human samples themselves were cultured. So essentially, we could generate what's called a BAC library, whereby some parts of the human genome are amplified, or I'm trying to think of the right term. A part of that genome would be within the bacterial cell, and the bacteria can be cultured and grown to make lots and lots of copies of those pieces of human DNA. And for the Human Genome Project, there was like a tile path of lots of BACs, and each of those were then sequenced separately. So effectively, the Human Genome Project, we sequenced it by breaking it down into lots of smaller manageable chunks rather than sequencing the whole thing at once, which back in the early days would have been quite a mammoth task. Simply the technology and the knowledge that we have now obviously wasn't there.
Yeah, sorry. Thank you, Nancy. And like you're saying, back then it was little fragments at a time. And then now the sequencing, the more modern sequencing, we can do tens of millions of fragments sequenced at once, which is a vast improvement in technology.
So I want to give our listeners a picture here of what happens to this initial sample that's taken. So I mentioned that there's one individual that is about 70% of the human reference genome, a little more when it was published in 2003. That individual has been given the name RP11. Wonderful anonymizing collection of letters and numbers there. But so we imagine, had it been our worm or our mice samples or our yeast samples, we can go back, we can get more DNA, we can do that in our labs. With RP11, once they've given that blood sample, that's it. They've gone. They've walked off. We can't get that back again. So that's where this process that Nancy's described there of creating this BAC library or this YAC library is really useful, because then we've got lots and lots of it. We can send to all these different labs around the world. They can do all of those steps that you've described there of sequencing. One at a go. Great. And I want to get us a picture of what that was like at the Sanger site during that time. So Nancy, you mentioned loading gels. I wonder if you could just give us a bit more of a picture of what was going on day to day when you came in on your Saturdays to help with the project. What did that look like? What did that mean?
Yes, certainly. So loading gels effectively is there were the sequencing machines have glass sheets, and you have to pour gels on them. And so there were lots of tasks effectively that needed to be done to get these samples onto the sequencing machines and then get the sequencing data off of the machines for analysis. And as I mentioned earlier on, there were quite a lot of opportunities for you to really put your hands to lots of different things. So I really enjoyed coming in at the weekends and loading the gels. It was an opportunity to, to contribute to this bigger effort as part of the Human Genome Project. And really, I think that there was a really good feeling and a good an amazing atmosphere at the Sanger Institute in the early days, as we're fortunate enough to have now. But there was a real group effort, a collective effort, that everyone would kind of muck in and all hands on deck to contribute to this. And, you know, if coming in at the weekends loading gels helped to make that happen, then it was really good to be a part of that.
Yeah, that's amazing, Nancy. And I guess, Liz, for you as well. So you were involved in sort of helping to integrate sort of like an innovative technology at the time. What was that like as well? That sense of this new grand project that where loads of people, as Nancy mentioned, that sort of collective aim to try and do this like amazing thing that had never been done before. What was that like, and sort of innovative elements of that?
Yeah, I mean, it was really exciting for me because I came from a background of analytical chemistry, really, rather than biology. So I came in to work in the R&D team and to introduce the new capillary sequencing. This was rather than these slab gels that Nancy's talking about that we have been working with, which are very manual and having to put samples in across the top of the gel manually and then look at the data manually. And there was a new instrument that could kind of automate this and really mean that we could ramp up and look at more samples more quickly. And also that the data analysis on that would be less manual. Now, this wasn't about cutting out the number of people working at all on it, but we were also in a battle. We were in a race because we were trying to get that genome released so that it was open to the public quicker than a private venture company were doing. So that feeling of all being in it together was there. It didn't matter whether we were introducing technology, because people were then like, oh wow, that's something new, let's have a look at that. Nancy's already described there were so many opportunities to be involved with different things that this was like the next thing. It's like, wow, and look how fast things are moving, and the speed of technology changes hasn't slowed up in that last couple of decades. Has that been that long, Nancy? and so that's why it felt exciting, it felt invigorating, and everyone just wanted to pull in together and do that.
And if I can quickly add to that, sorry, I think that one thing that really sticks in my mind is that race that you mentioned, Liz, and how it was really a massive collective effort. And I remember sitting, in a team meeting and my team leader saying to all of us, we all really need to think, what can we do to speed this up? Can you all think, can you any of you think of ideas and options that we can do to make what we're doing much more effective? And that was really empowering, and it was really exciting, like you say.
Yeah, definitely. And like you say, the weekend working, we were all in there doing that. And sometimes with that new technology that we had just introduced, we hadn't thought about those previous steps about making enough sample in a readable situation to fill all of that capacity now that we've got on that sequencing technology. So we then had to actually come in the week, on the weekends, to actually help make those DNA readable so that then we can actually keep that process going.
I think there's something really interesting here that almost changes the feel of our question when you start to talk about this race that it felt like the Human Genome Project was in with the private team that were working, because this whose genome was sequenced first, becomes, well, is it the open company who wants to publish it for everyone, or is it the private venture that would look to capitalize on some of the things that were discovered within it? So yeah, whose is it? Who perhaps does this genome belong to? And it's really interesting to hear. I think it's always great from both of you when we hear these kind of emotions that are going on within a team in that time. it sounds like it was quite a motivating force to go, we want to be part of the thing that publishes this for everyone. Just perhaps say a little bit about that, the openness of the data that was created and what that meant for the two of you.
Okay, do you mind if I go first, Nancy?
Yeah.
Actually, as one of my roles here at the Institute, I have been involved in data release, and I've worked in ethics and governance, and I was involved with releasing data, human data, because we have to be careful and sensitive, as we all know, that it is about a person's genes and understanding how their bodies work and everything. So I've been involved in that releasing of the data, and it is important that we do that, because when we're releasing that data, we're releasing it for the whole of the scientific community in an anonymized way, so that it can't be traced back. And it comes, and the human data, which has gone through participant and donor consent, which means that anyone who wanted to give their sample in for the research had to consent to everything and how the data would be used. Now that open release enables other scientists who work in drug discovery, maybe, and in a more clinical environment than we are. We are designed to create lots of data quickly. We do some functioning, understanding how the function of the genes work, but mainly that happens in other places, and as drug discovery, pharmaceutical companies and things. By making that data available, that's how we actually progress healthcare and improve drug discovery. So it's really important.
Yeah, and I think just adding to that, Liz, the feeling that you're part of this enormous groundbreaking project, and that actually it is for the benefit of everyone, or, you know, that's the aim and the intention. And it's turned out many years later that it has begun to revolutionize healthcare. It's led to so many advancements that we really hadn't thought about at that time. And I think when the Human Genome Project was happening, it felt special, but I think we perhaps really didn't appreciate just how special that project was. But I know that it definitely, I felt a sense of pride being involved in that project, and the openness of that data release, and the fact that it really could facilitate so much more research. I think that's a wonderful thing to happen, and it's great to have been part of that.
It definitely, and then, like you say, that pride thing, because it was something that no one even thought we could do. You know, no one had ever thought to sequence a human genome, and there we were, about to embark on this adventure. A bit like being an astronaut. Will we ever walk on the moon?
There was a very purposeful decision when they originally collected the samples to keep them anonymized. And there's a few different reasons why you might want to do that. But one thing was that they didn't want someone to be able to say, oh, it was mine. Mine was the first human genome. Look at me. This is about me. They wanted, the people who were making these decisions wanted it to be something that everyone could look at and go, that's something for all mankind, to continue with your astronaut reference. But there's another side to anonymization too, isn't there? And I'm interested to hear from perhaps you first, Nancy. How closely you linked the samples you were working with on the site with real people, or with a real person out there? What do you think are the pros and are the cons of that anonymization, meaning that it becomes a nameless liquid in a bottle?
The pros and cons, well, I mean, absolutely, that there are so many considerations around ethics when a human is providing a sample. And as our knowledge grows around what certain gene sequences might mean, it could potentially be used against somebody, in theory, you know, for it could be possible to learn something about somebody by looking at their DNA sequence, in theory. So it's crucial that it remains anonymized, and that's under the control of the individual providing those samples. And your question, Jack, about how close was I to, you know, when those samples come in, kind of, I guess, knowing where it came from. Those samples came in, and essentially one tube of DNA will look exactly like another tube of DNA, and they would be labeled with some, you know, you mentioned earlier on RP11, I think, and you know, the sample naming is incredibly it's not random, but it's very anonymized. It doesn't mean anything or relate anything in any way to the provider of those samples. I'm not sure if that's answered your question okay.
Yeah, I was struck by you saying one bit of DNA looks very much like another bit of DNA. I always think there's something kind of beautiful in that, because I think it is what unites us, isn't it? You know, I mean, often the research is trying to look for the differences and what the differences can tell us, but actually it's the overwhelming similarity, not just between you and I, but between me and that hedgerow that I mentioned out there, the DNA and that thing. Yeah. Liz, was there anything you wanted to add on that? What yeah, did you picture a person when you got a hold of a back sample?
So no, however, we did know that they came from participants, and we have the same care now as well over any of the samples that we have, and realise how precious they are. So we always work to not create waste or to waste any of that precious sample that has been given to us, because it's given to us. I've lost the right word, but for us to have responsibility and care and concern for that sample. And so we would always be respectful, and it's good that it's anonymized, and yeah, like Nancy says, that when we get them, we would treat all samples with that same level of care and respect anyway. But yeah, it's good to be anonymized.
Amazing. And I'm now going to shift us into a slightly future focused gear, but with just a very small caveat of just picking up on that thread that you mentioned, a little bit on that astronaut thread as well that Liz mentioned as well. The draft, the sort of Human Genome Project gets completed, and there's a grand sort of press conference with Bill Clinton and Tony Blair sort of presenting that. Like Jack said, we can put a video link to that in the description later. And you mentioned about that extreme amount of pride of completing this sort of groundbreaking project. I guess moving into the future now is sort of what's sort of next in both your areas, I guess, in terms of that was groundbreaking at the time. What's going to be the next potential groundbreaking thing in both your areas, do you think? Nancy, why don't you start with, because I know that Darwin Tree of Life project, that's another one of these enormous projects. Tell us a little bit about what that actually is trying to achieve again.
Yeah, I'd be happy to. So the Darwin Tree of Life project aims to sequence the genomes of all of the species that are found within the United Kingdom, within the British Isles. And at the moment, the approximate estimate for the number of species is 77,000. That number will undoubtedly change. But essentially, that means that we are doing the Human Genome Project to a greater or lesser extent on 77,000 different organism types, different species. So really, the challenge for us is to be able to do that in a really high throughput manner. You think about the number of years it took to do the Human Genome Project. We're now trying to do that 77,000 times and do it well.
Yeah,
that's an amazing challenge.
Amazing.
That sounds like a lot of Saturdays coming into those gels, Nancy.
It does, yeah.
What allows you to do that that big scale?
Yes, fortunately, we've gone past that. So the technology's moved on so that we now can sequence longer pieces of DNA in one go, which means that putting it back together again into understandable sequence of DNA letters is much easier. It's still not easy, but it's much easier than it was back in the early days. And also in terms of scale, so we're constantly looking at technological improvements that teams like Liz's can then put into place to operate not just on one sample at a time, but 96 or 384 samples at any one time to really give us that sense of scale.
It sounds like Nancy's sending you a lot of samples then, Liz, from the sounds of it. Yeah, Liz, tell us, something I wanted to pick up a little bit earlier, but I guess looking into forward, Nancy's just mentioned 13 years to sequence one human genome. Tell us, yeah, what are the next steps for you as implementing new technology and stuff like that? But how quick are we doing it right now?
Oh, rapidly. I mean, the initial plan for the human genome was that it was going to take at least 15 years. Because of adding in technologies like the one that I was helping with when I started, we were able to bring that down to be able to release the draft in 2000. And then we closed up some more gaps because there were some bits of data missing so that we could actually have a finished draft after 13 years. But no one thought that we would get the bulk of it done in 10 and then really closing that up together and making it a publishable genome within the next 3 years. So that's how quickly technology is moving. And now technology is moving so fast, we can sequence I think we're now close to 80 human genomes in 48 hours. Okay.
Okay, wow. And
so this is using a slightly different technology to the
Yeah.
longer pieces of DNA that Nancy is talking about. But my team, because we look at much smaller pieces of DNA and stick them together in a very similar way to those earlier days, it just means that we can read tens of millions of those small fragments at a time now. And the technologies that Nancy's using in her team with the Tree of Life, those technologies are going to be starting to embrace a lot more automated, which means that a robot, we call it a robot, will move samples around instead of humans because they can do a lot more in a short amount of time.
Amazing. It sounds, yeah, I can't believe that stat of 80 in 48 hours. That sounds, yeah, that's just crazy amount of time. Yeah. And I guess for you, it's been really nice to sort of see that evolution happen, I guess, and be a part of implementing new technology each time that it comes into play.
It really has been. It's been really, really good to look at that. But not only the technologies now that we're using that are changing, but the way we make that DNA readable is changing because we're reading different parts of DNA. Because there's other molecules that are stuck on that DNA, and we're reading those now so that we're understanding other things which might affect how genes work. So it's actually the deeper understanding of the biology now, not just here's a string of letters, but actually what else? Is there like a little subscript? Is there another little bit of a note stuck on the side of that? And what does that tell us about how that gene's going to work?
Amazing. Well, I'm just looking at the time and we have flown through this session. So I just want to first and foremost say both a massive thank you to Nancy and Liz for joining us today. I would also like to thank everybody who's watched wherever you are in the world, and just give a quick nod to next week's session. Nancy and Liz have both mentioned about sort of sequencing, short read sequencing. What we're actually going to be looking at next week is about what's got the biggest genome. So we're going to be looking at what species have the biggest genomes. We're going to be looking at long read sequencing that is doing a lot of the stuff to do with the Darwin Tree of Life project and the way that they're starting to piece those large genomes together. But yeah, any final words from you, Jack?
Just to say once more, thank you very much, Nancy. Thank you very much, Liz. And we're looking forward to seeing you next time.
Thank you very much. And if you want to join us for that next episode, all the details are on YouTube. And yeah, please do click that notify button if you want to be informed just before the session starts. But yeah, it's been lovely having you all. And again, thank you, Nancy and Liz.
Yeah.
Thank you.